Hello, my name is Anne-Marie Winger. I'm a board member of the European Society of In Vitro Toxicology, and welcome to our March webinar here in an ongoing series presented by ESTIV and the American Society for Cellular and Computational Toxicology. I encourage you to visit our websites to find out more about our organizations. And please consider becoming a member to support programs like this webinar series. You can also join our mailing lists for job and event announcements. This webinar will be recorded, and the recording will be hosted on the ASCCT website a few days from now. In the webinar platform, you can ask questions to the presenters using the Q&A feature at the toolbar at the bottom of your screen. There'll be a Q&A at the end of both speakers' presentations. So add your questions at any time, and other participants, they can see and vote for questions they really want to hear the answer to. And please indicate which speaker your question is directed to. And finally, please say hello in the chat and let us know where you're joining from. And then I have a few announcements from the societies. The 12th annual meeting of the ASCCT will be held in the Washington, D.C. area from the 23rd till the 25th of October this year. And the first 2 days will be the meeting program, and the 25th will be a set of courses. And the submissions is currently open for course proposals and general presentation abstracts. Please use the QR code on your screen or go to the ASCCT website to learn more. Then the Physicians Committee for Responsible Medicine in the US is seeking applicants for an open position within regulatory science policy. So please go to the PCRM website to learn more and eventually apply. And then registration is now open for the next webinar in the Physicians Committee's NAM use for regulatory application dynamic discussion series. And this webinar will be held on the 14th of April from 10 a.m. Eastern Time, and it'll cover new strategies for safety assessment of endocrine disrupting chemicals. So please scan the QR code or visit PCRM website for more information. Then ESTIV is arranging a training course in vitro toxicology. It's a 6-day course, and it's taking place from the 29th of May till the 3rd of June this year at the Slovak Academy of Science in Bratislava. And the course is open for registration. So please take a look at the ESTIV website for more information on that course. Then the next joint ASCCT and ESTIV webinar in April is also open for registration. We will hear from 2 presenters from the 22 ESTIV annual meeting on the modeling of adverse outcomes. So please register through the ASCCT website. The registration information will also be shared here in the chat. And today's webinar is on the use of new approach methodologies for evaluating cardio and developmental toxicity. Our speakers both won awards for their presentations during the 22 ESTIV annual meeting, and we are so excited to have them present for you all today. Our first speaker is Dr. Erika Ferrari. She obtained her PhD in bioengineering at the Politecnico di Milano in 2022, with a thesis focused on the development of liver-based multi-organ-on-chip platforms as advanced in vitro tools to predict drug metabolism effects. She recently joined BiomimX, where she currently works as a business developer. Today, she'll present her latest work focused on the development of a multi-organ chip platform to study off-target cardiotoxicity of drugs upon liver metabolism. Our second speaker is Yanying, who has a master's degree in environmental chemistry, toxicology, and human health from the University of Copenhagen. And currently she is a PhD student at the National Food Institute at the Technical University of Denmark, where her focus is on predictive and mixture toxicology of chemicals. And today she'll present her latest work on the effects of pyrethroids on differentiation of human-induced pluripotent stem cells. So this was all for me, and now I will give the floor to Erika. Please share your slides, Erika.
Yes.
And
can you see?
Yes.
Okay.
It's all clear.
So thank you, Anne-Marie, for the nice introduction. And thanks to the organizers for inviting me to present my latest work at today's webinar. And as Anne-Marie was anticipating, today I will talk about a multi-organ-on-chip platform for the in vitro investigation of target cardiotoxicity of liver-metabolized anticancer drugs. Organs on chip are microfluidic devices able to mimic activities, mechanics, interactions, and physiological responses of human organs in vitro. They hold the great potential to be applied in the drug development pipeline for what concerns efficacy and safety to complement and eventually replace animal models. In fact, animal models are still the gold standard, but there are several differences between animals and humans for what concerns disease pathways and drug metabolism. There is also animal-to-animal variability and relevant ethical problems. So animal models are not sufficient and need to be complemented with in vitro human studies. In fact, 2D and 3D cell culture can be used with the human cells, and they are cheap, but they are not able to recapitulate the intrinsic organ complexity. So organs on chip have been developed and they are able to host low volume samples and being still with low cost. And within the organs on chip, human cells can be positioned in a controlled environment in 3D, so to better recapitulate organ architecture. And also different stimuli can be applied to the devices, so to better mimic organ complexity. But a single organ on chip is not able to recapitulate different organ districts such as what we have in our body. So more recently, multi-organs on chip have been proposed, and they are able to recapitulate the interconnection of different organs, so to control and understand the communication, and so to also early eliminate problematic drugs. In the context of drug safety, the two most targeted organs are the heart and the liver. In fact, when an anti-cancer drug is administered in the body, it is firstly metabolized by the liver before reaching the tumor target site, and the produced metabolite may reveal unpredicted toxicity. And in particular, off-target cardiotoxicity is the leading cause of drug withdrawal from the market. This field is still evolving, as the few platforms available encompassing liver and heart co-culture still need high priming volumes, or they use monoculture or 2D culture with low functionality, and also there is no control on organ-organ communication. In our lab, we developed a liver and a heart model. The liver model is based on the micropattern co-culture model of EBG2 and fibroblast, and it was optimized for microfluidic platforms and validated for metabolism. Whereas the heart model is a 3D model where neonatal rat cardiomyocytes are embedded in a fibrin gel and seated within the device. And the platform is capable of mechanical stimulation and electrical recording, so to enhance cardiac viability and improve maturation and functionality of the cells. I'll go now a little bit deeper within the two single models. For the liver model, we started, as I said, from the micropather culture model developed by Ketani and Bhatia, where hepatocytes are arranged on a circular domain of collagen surrounded by fibroblasts. And within this configuration, the hepatocytes are able to produce a high level of albumin compared to when they are cultured alone. But this model was mainly used at the micro scale. So during my PhD, we developed a technique to incorporate the MPCC within sealed microfluidic devices, taking advantage of the plasma exposition and combining patterning and microfluidics, so to obtain circular collagen domain within sealed microfluidic devices that could be used for cell culture. Within the device, we successfully seeded FG2 cells on the circular collagen domains, and also 3TT fibroblasts could be seeded all around the hepatic islands. Additionally, the cells maintained high viability for 1 week of culture, and the hepatocyte could express good albumin levels. The liver model was also validated for metabolism, and in particular, we used the anti-cancer drug Tegafur, which is non-toxic until it is metabolized by the liver into 5-fluorouracil, which instead is toxic for colon cancer cells. As you can see from this graph, when Tegafur is firstly metabolized by the liver, there is a decrease in tumor viability following the trend of the already metabolized drug 5-fluorouracil. And if you want to know more about this specific project, have a look at the bottom publication. For what concerns the heart model, the first integrated technology is called UBIT, and it's a mean to provide a controlled mechanical stimulation to the cells embedded in a fibrin gel, here represented in blue. And for the heart in particular, we are able to provide a uniaxial stretch of 10–15%, so to generate functional 3D construct. After 1 week of culture, the cells appear interconnected, and as you can see from the immunofluorescence staining, the cardiomyocytes in green are able to express cardiac troponin I. And from the magnification, you can appreciate the fact that the cardiomyocytes maintain the typical striation of the sarcomere. Also, the construct is beating in a synchronous way, and 7 weeks of mechanical stimulation also enhance the expression of ion-related genes, such as calcium, sodium, but mostly the potassium genes. The second integrated technology is called the micro-ECG, and it's a mean to precisely position an electrode in close contact to the beating microtissue, so to both control the electrical stimulation that we can apply to the cells, but also to monitor the electrical activity of the construct. And as you can see from this image, we are able to record the signal in different parts of the microtissue. For example, here we have two signals, yellow and blue, that are acquired at the two opposite ends of the microtissue. And the signal overlap perfectly, meaning that the construct is beating in a synchronous way. And we can appreciate the spike, which is the polarization, and the smooth wave, which is the depolarization. And from this, we are also able to capture the typical electrophysiological parameters, such as the beating period, the field potential duration that we also correct with the Friedrichs method, and the amplitude of the spike. So we started from these two models, the liver and the heart model, and we developed a multi-organ-on-chip platform capable to detect the cardiotoxicity of drugs upon liver metabolism. The platform is called the LivHeart, and it is composed of a chamber layer where we see two different compartments, one for liver and PCCs and one for the 3D cardiac construct. In particular, in the heart chamber, we see 4 guides that are needed to insert the electrodes, so to eventually record the cardiac electrical activity. On top of the chamber layer, we have the valve layer. In particular, we have 2 valve systems. The reservoir valve system that is needed to isolate the liver chamber from its reservoirs, and the communication valve system that instead controls the communication between the liver and the heart chamber. In particular, these are normally closed valves that can be opened when a negative pressure is applied. On top of the valve layer, we have the actuation layer, which is needed to provide a uniaxial mechanical deformation to the cardiac cells embedded and seated in the chamber layer. And this is a picture of the assembled device. Firstly, we assessed the operating pressure of the valve layer. So we inserted a colored PBS within the valve system, and we applied the decreasing pressure values. We analyzed the fact that the communication valve system could be open when a negative pressure of 550 millimeters of mercury was applied, whereas the reservoir valve system could be opened when a negative pressure of 200 millimeters of mercury was applied, and both these pressures are compatible with the biological lab supply. Then we assessed the operating pressure of the actuation layer and the stretching that we were able to generate within our cardiac chamber. To do so, we injected a suspension of polystyrene beads embedded in fibrin gel within the central channel of the heart chamber. And by increasing the pressure that we were applied to the actuation layer, we analyzed the movement of these beads, so to correlate the movement to the strain that we were able to obtain within the channel. And we validated the fact that we were able to obtain 10–15% physiological uniaxial strain within the heart chamber. Then we assessed the diffusion behavior that we were able to obtain within our device through the software console. And for instance, by administering a 10 micromolar concentration drug within the liver chamber, after 2 days, we are able to obtain 3.15 micromolar concentration of the same drug within the heart chamber. This was also validated experimentally, and we introduced a suspension of polystyrene beads and rhodamine, which is a dye with a molecular weight similar to the drugs that we want to use in our study. And we administered this suspension within the liver chamber. And by opening the communication valve system, we monitored the movement of the beads and of the dye towards the heart chamber. And as you can see from the picture and the graph, we can see that the rhodamine slowly diffused from the liver towards the heart chamber, whereas there is no beads movement, meaning that we are able to control the diffusion within the device, and the device is capable of avoiding convection. From the biological point of view, we successfully obtained functional micropattern co-culture of HBG2 and fibroblast within the liver chamber, and the hepatocytes were able to express cytoplasmatic albumin, as you can see from the immunofluorescence staining on the bottom left of the slide. And the cardiomyocytes were able to synchronously beat And also they express the typical markers such as troponin I and connexin 43. Finally, we studied the behavior of a drug within our model, and we used terfenadine, which is a cardiotoxic drug that is toxic for cardiomyocyte until it is metabolized by the liver into fexofenadine, which instead is safe for cardiac cells. Additionally, terfenadine causes the prolongation of the QT interval of the cardiomyocyte. So after the obtainment of the two cell construct separately, we administered terfenadine in the liver chamber. After 24 hours, we open the communication valve system. And again, after 24 hours, we analyze the electrical activity and the viability of the cardiomyocyte. Firstly, we wanted to make sure that the liver model was able to really metabolize terfenadine to fexofenadine, and we ran a mass spectrometry analysis. And as you can see from the picture, fexofenadine was detected at 2 minutes, whereas residual terfenadine was detected at 2.48 minutes. And in 5 replicates, we validated the fact that 90% of terfenadine was correctly metabolized into fexofenadine. For what concerns the viability results, we can see that when terfenadine is directly administered on the heart, there is a decrease in the cardiac cells viability, whereas this effect is limited when it is firstly metabolized by the liver, following the trend of the already metabolized drug fexofenadine. Similarly, for the liver model, we just checked that terfenadine wasn't causing a decrease in hepatic viability. Concerning the functionality results, we continuously monitored the electrical activity of the cardiac construct, and by monitoring the beating period and the field potential duration. And as you can see from the graph, when terfenadine is directly administered on the heart, there is an increase in the field potential duration as expected, whereas this effect is again limited when it is firstly metabolized by the liver, following the trend of the already metabolized drug fexofenadine. So the LivHeart allowed the testing of a non-cardiotoxic metabolite generated from a cardiotoxic drug. To conclude, we designed and developed a reliable micropattern LivHeart platform encompassing a continuously monitored mechanically active 3D cardiac model to undertake drug toxicity studies upon hepatic metabolism. And we demonstrated the case study terfenadine. Of course, the model still has a limitation because the liver chamber is still 2D, the device is in PDMS, user-dependent with low to mid throughput. But in the future, we would like to switch from a 2D to a 3D liver model, and also adopt human-derived and the hepatocyte as more relevant cell type to be applied within the LivHeart platform. So now I want to thank my lab in Politecnico di Milano, BiomimX, our collaborators, and again, the organizer for inviting me today. And thank you all for your attention. And at the end of the webinar, I will be pleased to answer any question you might have. Thank you.
Thank you, Erika. Thanks for a nice presentation. And please remember to ask questions in the Q&A function. Then we'll discuss afterwards. So now it's Yanying's turn to give a presentation. Please.
Yes, I'll just start to share my screen. Can you see my screen?
Yes, we see it.
Yes, okay. I would like to first thank ASCCT and STIF for giving me this opportunity to present some of my work on pyrethroid insecticides using assays that's established by some of my colleagues here at DTU. I will first start to introduce you to developmental toxicants. Let me just have my pointer on, yes. And before knowing what is a developmental toxicant, we first need to know a bit about the human prenatal development. Human starts with a fertilized egg on day 1, and after several runs of cell division, forms the blastocyst on day 4. And the cells in the blastocyst will form the inner cell mass, and which is the first step to form the 3 germ layers and becomes an elongated structure. And we will first see the beginning of heart development with the mesodermal cells migrating to form the primary and secondary heart field. And on day 22, we will have our first heartbeat, and all organs will be developed until week 9, where the embryo becomes a fetus, which continuously grow until birth. And pregnant women during this sensitive period of time can be exposed to different substances and chemicals. And these substances, if they can cause birth defects or other type of abnormalities, they are called developmental toxicant. And I believe many of you know Agent Orange, which is a really good example of developmental toxicant. But for some that might know much about this herbicide, it was used in Vietnam War between 1960s to 1970s. And it's a mixture of 2 different herbicides. But during the production of Agent Orange, TCDD, which is one of the most toxic dioxins, are produced as a byproduct, and it's not avoidable. And the most predominant effect that's linked to exposure to Agent Orange was spinal cord defect. And there's another very famous developmental toxicant, which I believe a lot of you know about. It's thalidomide. It was prescribed to pregnant women in the 1960s to treat morning nausea. It was tested safe in rodent studies, but half of the babies were dead prematurely, and the other half were born with limb defects and similar. And because of thalidomide, rabbits were introduced as a model to further test developmental toxicity. And here is a pie chart and the table showing you animals used in the EU in 2019 for testing developmental toxicity for regulatory use and routine production. And we can see that around 83,000 animals were used in 2019 alone in the EU. And out of these 80,000 animals, rats and rabbits were the most used animal species, and they together account for more than 90% of all animals used in developmental toxicity testing. And just by looking at the animals used for toxicological purposes, rats and rabbits were mostly used for reproductive and developmental toxicity testing. However, with increased amount of chemicals that needs to be tested and growing concerns about animal welfare, as well as increased awareness of limitations of animal models, there has been a shift towards developing new approach methodologies that follows the 3R principle, which is also a firm legal requirement that's implemented throughout the EU directive on the protection of animals used for scientific purposes. And here are some in vitro and ex vivo approaches already existing for assessing developmental toxicity of chemicals. On the top two, we have assays that use whole embryo culture derived from rat or zebrafish. And on the left and right, we have models that's using embryonic stem cells derived from mouse or human. And we also have a relatively new assay, ReproGlow, which is a reportaging assay that's using mouse embryonic stem cells. But if we look at all the assays here, most of them have ethical issues, and many of them are not human relevant. So we really need assays that has higher throughput potential and of human relevance. So we don't need to extrapolate between different species. And all these in vitro assays or the new approach methodologies hold promise for providing more accurate and ethical assessments of risks associated with chemical exposure. And this brings me to my next slide, which is the use of human-induced pluripotent stem cells, HIPS, as a model for embryonic development. Traditionally, we can only isolate the pluripotent stem cells from blastocyst stage embryo or fetus, which has a lot of ethical issues. But thanks to Shinya Yamanaka and his team at the Kyoto University in Japan, who in 2006 identified 4 transcription factors when introduced into mouse fibroblast cells, could reprogram these cells to induced pluripotent stem cells. And in 2007, they were able to repeat this fate with human fibroblast cells. So now we can not simply, but we can easily reprogram somatic cells from human adults into human-induced pluripotent stem cells. We have taken advantage of using embryonic bodies, EBs, in our lab, and they are aggregates of HIPS that mimics the very early stage of an embryo, the blastocyst. And EBs can be differentiated into most cell types of the body, including cardiomyocytes. And our previous PhD student, Karen Lausch, has developed this assay called the pluribeat, where on day -1, we seed 5,000 cells into a conical shaped 96 wall plate, and we centrifuge the plate down for the HIPS to aggregate to become EB by day 1. And then for the next following days, we expose EBs to different differentiation medium. And these mediums composition are relatively complicated, but you can find more information in the references that I included at the bottom of the slide. And by day 7, we have beating cardiomyocytes. And here are a series of images showing you the development from EB on day 0 to day 7 beating cardiomyocytes. And when we are looking at the compounds of interest, we just expose EBs to different chemicals, different concentrations at the days indicated by the red arrows. And it's also important to notice that this is a relatively long protocol, 7 days, but we can free. And on day 7, we would beat score the contraction of cardiomyocytes, and we give them 3 scores. 2 means fully beating, and 1 means partially beating, and 0 means no beating. And here's a video showing you day 7 beating cardiomyocytes. I first need to, yes, now I can show you the video. Yes, and I have to put my pointer back again. Yes, it is very important to analyze if the differentiation of EBs truly follow key developmental stages of the developing embryo in humans. Therefore, a set of markers were analyzed. So the T gene here, it's a mesoderm marker. And you can see that it was induced on day 0 and peaked on day 1 and decreased again. And this confirms the formation of mesoderm. And the ISL1 and MESP1, which is early cardiomyocyte marker, was increased in day 1 and stayed more or less the same in the following differentiation days. And then NKX 2.5 was increasingly expressed following the differentiation day. And last but not least, all the late cardiomyocyte markers were increased from day 3 onward and peaked at the last 2 days of the differentiation. And we also looked at the expression of pluripotency markers, and you can see that they were all downregulated when we were differentiating from EBs to cardiomyocytes. And this data together confirmed our findings that the EBs differentiated into cardiomyocytes through developmental stages that also occur in the human developing embryo. And to get a better understanding of the distribution of cardiac cells within the spheroids, we also investigated the cardiac marker proteins across the EBs using immunofluorescence staining of NKX 2.5 and CTNT, which is cardiac troponin and is encoded by TNNT2 gene. And the picture D is a zoomed in version of C, and you can see DABI stained for the nucleus, and NKX 2.5 is mainly presented in the nucleus area, and CTNT is mainly presented in the detected in the cytoplasma area. We therefore concluded that our protocol ensures a robust and reproducible differentiation into cardiomyocytes. Now, if you still remember this slide, the pluribeat assay, since we have to beat score all our plates on day 7, which is a time-consuming process, and it also introduces potential biases because it's very subjective. My colleague Andreas Tresco and the Danish company BiomimX used CRISPR-Cas9 technique and inserted nanoluciferase gene into the NKX 2.5 gene. So on day 7, after beat scoring each plate, we then measure the luminescence readout using a luminator by adding nanoluciferin into our plates. And this allowed us to have a higher throughput potential assay. And the changes in luminescence readout is an indirect measurement of changes in NKX 2.5 expression. Oh, and also to confirm the ability of the cells to form derivatives of the 3 germ layers, we differentiated the gene-edited HIPs to cardiomyocytes again, and it showed that the high proportion of the cells expressed markers for the respective germ layers, and the HIPs line were also characterized after the insertion of nanoluciferase gene, and more than 99% cells were positive for pluripotency markers. But it's not shown here, but if you are interested, you can find more information in the references that I included at the bottom. For our project, we chose pyrethroids based on that they were the most often detected pyrethroids in Danish foods, and we also included the common metabolite. And because pyrethroids are known neurotoxicants and they act primarily by interfering with voltage-gated sodium channels, which are also essential for cardiac muscle contraction, we therefore hypothesized that them to be cardiac toxic, but we do not know much more about it. Therefore, we wanted to evaluate the potential developmental toxicity of pyrethroids and the common metabolite using a stem cell-based assay. So here are some results from our lab tested by my colleague. And on the top left corner here, we have one of the PFAS. You can see that it negatively affected NKX 2.5 expression and the contraction of cardiomyocytes. And the fungicide here only negatively affected the luminescence readout, but not the bit score of cardiomyocytes. And one of the vitamin here had negative results from both of the endpoints. And before showing you the results on pyrethroids from the pluribeat and the plurim assay, we first established non-cytotoxic ranges by exposing hips in monolayer to different concentrations of chemicals for in total of 48 hours. And we measured cellular ATP level and set the cell viability cutoff to 80%. And here are the results on pyrethroids and the metabolite from plurilum and pluribeat. It's a lot of figures, but I will summarize the information here. As you can see, all the pyrethroids negatively affected the NKX 2.5 expression at a relatively low concentration, but only the PYT2 affected contraction of cardiomyocytes as well, not the other two pyrethroids. And the metabolite had no effect on either of the endpoint at up to the highest tested concentration. So there are some take-home messages and future perspectives from my presentation today. We know that the pyrethroids negatively affected cardiomyocyte differentiation in the plurilum assay, but not the metabolite. And the plurilum is more sensitive compared to the pluribeat assay for the pyrethroids that's tested in our lab, but we can also say this for many other compounds that have been tested. And so far we have tested somewhere around 15 chemicals, but we also need to expand our database and test more chemicals. And we also know that the human exposure levels are predicted to be much lower than the tested concentrations, but we do not know what's the effect when the chemicals are in a mixture. That's also one of the future perspectives that we're planning to work on. I would like to thank my colleagues from Technical University of Denmark and our collaborators from University of Southern Denmark and the Danish company BiomimX and the grant provided by Danish EPA. And thank you everyone for listening. I would be happy to answer your questions later.
Thank you, Yanying. And thank you to both of you for nice presentations. There are a few questions in the Q&A. The first one is, I think it's for Erika, how to select the cells for developing the organ on a chip.
Okay. Yeah, I would say that what we do, usually we start from cell lines because are more characterized, so we are better able to understand the behavior of the cells within the device based on the stimuli that we are given. But for instance, once the model is settled with the cell lines, we try to move on more relevant cell types such as primary cells or iPS. And for instance, for the heart, we started from neonatal rat cardiomyocyte, but we are now working on human-induced pluripotent stem cells derived cardiomyocyte. So it's always also a way to stay cheaper at the beginning with cell lines, and then once the model is consolidated, move on more relevant cell types.
Thank you, Erika. Yeah, and then there's a comment, I think. It's just to clarify the developmental toxicity assay was developed with human embryonic stem cell, but also uses induced pluripotent stem cells. Almost all studies using this assay are currently being conducted with human-induced pluripotent stem cells. Do you want to comment on that, Yanying?
Yes, I would just like to say thank you for this information. I did not dig that much into the ex vivo and in vitro assays that I presented in my presentation. But yes, thank you.
And then there is a third question. My question is how 3D embryonic model is clinically relevant to demonstrate compound-related teratogenicity and embryonic fetal toxicity. That is for you, Yanying, I guess.
Yeah. Yes, I think this is a very good question, but also a big question. But the whole purpose of using embryonic body is that because it represents the early stage of an embryo, the blastocyst. And we, in the end, measure the expression of NKX 2.5 gene, which is a key early cardiac transcription factor. I think that would more or less answer the question, but it's also a very big question. I have to admit that I do not have a lot of expertise in this field.
We can also say that the heart is the first organ to develop in the body.
Yeah.
And so the hypothesis is that these embryonic bodies that mimic the blastocyst will be able to illustrate toxicity at on a very early stage embryo. Yes, and please ask more questions if you have any out there. Let me see. Maybe there's one more actually. It's for Erika. What is the advantage of diffusion over flow for linking the two chambers?
Yes, thank you for the question. So we prefer diffusion so that we can really control the movement of the metabolite rather than giving it all at once to the heart chamber, because with the diffusion, the liver keeps metabolizing, whereas if we give convection flow, the liver stops the metabolization that is doing, whereas with the diffusion, because it has more time, it metabolize it and also still start to go within the heart chamber.
Okay, thank you. Then there's another question here for Yanying. Have you tested other chemicals using the plurilome assay, or do you plan to?
Yes, if you remember the two articles that I have included at the bottom of some of my slides, there are already chemicals that's tested and included in the paper. But my colleagues are also testing more chemicals, and it's something to be expected to be published this year. And in total, we have been testing around 15 chemicals. Yes.
Yes. Okay. Let's see. Yeah, for Erika, there's a question in the chip system. Do you foresee challenges with certain compounds, such as high log P?
I am not familiar with this compound, but for sure, as we are using PDMS, probably for some molecules there can be binding of some molecules. So this is also why everyone in this field is also trying to move on more different materials like plastic materials, or they are trying to find strategies to pre-coat the PDMS so that it doesn't absorb lipophilic molecules. So yes, this is something everyone is facing in the field.
Thank you. Another question for you, Erika. How many chemicals have you tested using the liver and cardiac organ on chip model that you demonstrated in your presentation?
Within this specific project, we only tested the terphenadine. So only this compound. We are foreseeing to test others such as Tegafur also, but it's still an ongoing process.
Thank you. And for Yanying, could you please explain the steps that you follow to create the embryonic models?
I think that what you mean is the creation of embryonic bodies. We just harvest the human-induced pluripotent stem cells on day -1, and then we seed 5,000 cells in the conical-shaped 96-watt plate, and then we would centrifuge them and leave them in the incubator for a night, and by the next day they would form embryonic bodies. And then we would change medium, as I have shown you in my presentation. But if you want even more detailed explanation, I can perhaps attach the 2 papers that I have included in my presentation in the chat, so you can look it up.
Thank you. And another question for you, Yanying. Thanks for the very nice presentation. For chemicals like pyrethroids, which are very lipophilic, the dose to the tissue may be much higher than the dose administered to the aqueous medium. Therefore, in vitro to in vivo extrapolation should be considered.
Yes, I think that would apply for all in vitro assays. And this is something definitely for us to consider, and even comparing the concentrations that we are using in Petri dish compared to what we are exposed to in human blood. There is a differences, but that's a good point. But we have not come to that step yet.
Thank you. And I think this was all the questions. Let me see if there's anything in the chat. I think this was it for now. Yeah. So yeah, I would like to thank both of you for some nice presentations, and I hope that you will enjoy the weekend that is coming soon. So thank you for today, and thank you to all of you who are joining online. Bye-bye.
Thank you.